The cell wall is a layer outside of the cell's membrane that not all cells, for example animal cells, have. It is strong and rigid so that it may maintain the cell's shape and structure as well as protect the cell from outside environments and internal turgor pressure. These functions are especially clear considering extremophiles, cells that live in extreme conditions. There are psychrophilic cells that live in cold temperatures, thermophiles that live in hot temperatures, halophiles that live in salty conditions, and more. The thermophilic archaea and bacteria in pictures below produce the color seen in the hot springs of Yellowstone Park. Bacterial cell walls are made primarily of peptidoglycan, molecules formed from carbohydrates linked by peptides. This picture of Yersinia pestis, which causes the bubonic plague and does have a peptidoglycan cell wall. However, chlamydia, for example, is a bacteria which has a cell wall not made of peptidoglycan. Pyrella and Isosphere are others which have proteins instead to help give structure to their cell wall. There are some bacteria that have no cell wall at all which give them the suffix plasma, for example mycoplasma. Gram-positive bacteria have a thicker cell wall than gram-negative bacteria, containing about 80% peptidoglycan. Gram-negative cells have a second lipid bilayer outside their cell wall, making them harder to treat with antibiotics. To remember which is which, think of this acronym, long PowerPoint, like so many lessons from school. LONGPPT. Liposaccharide, outer membrane, negative, glue, as in stronger, harder to penetrate, than positive, peptidoglycan, thick. Archaea cell walls are made of pseudopeptidoglycan. In archaea and bacteria, the cell wall acts as a surface for proteins and bacteriophages to adhere to. Enzymes, for example, bind to specific domains that facilitate the cell wall disassembly by hydrolysis. Understanding more about the cell wall and its binding domains can aid the development of oral vaccines and serve as biomarkers so that scientists can identify the industrially important methanogen archaea. Plant cell walls are primarily composed of cellulose. Plant cells begin with a thin, flexible cell wall known as the primary cell wall. Upon maturation, plant cell walls develop a multi-laminated layer known as the secondary cell wall between the primary cell wall and the plasma membrane. It provides greater durability. Between the cell wall is a thin polysaccharide layer known as the middle lamella, which holds the cells together by sticky peptides. Cell walls have gaps such as plasmodesmata in plant cells so that they may facilitate cell-to-cell communication. These channels let cytoplasm of one cell run into that of another, unifying the entire plant. The plant cell wall forms during cytokinesis of an existing plant cell. The cell plate forms down the middle of the cell where cell wall materials gather, splitting to give a new cell wall for two daughter cells. When a plant cell is in a hypotonic solution that contains less solute than the cell's internal concentration, the central vacuole becomes filled with water and pushes against the cell wall to make it turgid. In an isotonic solution, the concentration outside of the cell is equal to the concentration inside the cell. This makes the cell flaccid. When a plant cell is surrounded by a hypertonic solution containing more solute than its internal concentration, the cell experiences plasmolysis, the retraction of the central vacuole from the cell wall and the constriction of its contents. If the cell did not have a cell wall in a hypotonic solution, it would lyse. Remember, salt sucks. Water will flow to where the solute is. It is important for a plant cell to be in a hypotonic solution because it keeps the cell turgid and maintains a plant structure. The cell wall can be altered to support mutualistic relationships between two organisms in a process called symbiosis. Symbiosis can be vital to an organism's survival, such as by allowing for developmental coordination, nutrient exchange, and protection against pathogens. It is important to distinguish whether the plant or bacteria is degrading the plant cell wall because it shows which one is promoting the communication. In order for a microbe to invade the plant cell, localized softening would be required. The degrading or weakening of interactions between plant cell wall polymers or lytic agents, either in the host or microbe. Plant cells may contain these lytic agents. It can stop or promote interactions between itself and other organisms. Fungal cell walls are made of chitin. Chitin is largely composed of nitrogen-containing monomers joined by glycosidic linkages. Plant roots and soil fungi, known as mycorrhizae, have symbiotic associations. Fungi have different carbohydrate activity enzymes that degrade plant polysaccharides materials to facilitate infection and/or gain nutrition.